Okay, this is the last set of notes on evidence for evolution, phylogeny or cladistics, and also the origin of life. This encompasses 2 chapters and then part of another chapter, so we're going to go really fast because there's a lot to cover. Remember that a theory in science accounts for many observations and explains and integrates a large variety of phenomena. It is more than a hypothesis, but again, it is a theory, and theories do change. They are based on evidence. They are not facts. They are not proven. We can't prove things in science. We can only say the evidence supports something. Macroevolution is a pattern of evolution over time that links all living things to a common ancestor. You're going to see that term common ancestor fairly frequently in the coming slides. So remember that Darwin had a great deal of evidence supporting the concept of descent with modification or natural selection. But he admitted that there were instances where key evidence was lacking. In the last 150 years, new discoveries have filled in a great deal of those gaps and continued to today. 4 types of data that document the patterns of evolution, so the macro here, are direct observations that we can make, homology, the fossil record, and biogeography. So we're going to talk a bit about each of those areas. The direct observations, for example, bacterial resistance. We talked a little bit about that. MRSA, methicillin-resistant Staphylococcus aureus, which is a staph infection that is resistant to known antibiotics. It used to be 50 years or so ago, we could prescribe penicillin for a staph infection, and it worked just fine. We now know that penicillin might cure strep throat at best. So this is an example of natural selection that we can actually observe because bacteria reproduce so quickly. The peppered moth, if you remember this one from Bio 1 during the Industrial Revolution, the color of the moths went from a kind of a lighter color to a darker color because there was so much soot on the trees that then the darker colored moths blended in better, survived, reproduced, and passed on their darker genes to their offspring. Silkberry bugs, I'll show you this here in a second. This is a chromosomal map, methicillin resistant. They've actually identified this as a gene and its placement in the genome of a typical bacteria, or the Staphylococcus bacteria, SARS. And it also has other genes that they've identified in there, but this is the one that is resistant to methicillin, which used to be the best known antibiotic for staph infections. This is the silkberry bug, so they actually manipulated this bug and they changed its food source on purpose, and they noticed that there was a big shift in the beak length for that particular insect population. Homology is another piece that we talked about, and this is where related species can have characteristics that have an underlying similarity, yet they function differently, and that we're what we call homologous structures. Remember that term. This similarity in structure results from a common ancestry. The view of evolution as a remodeling process leads to the prediction that closely related species should share similar features, and they do. Decidual structures are remnants of features that served a function in the organism's ancestors. For example, snakes do not have legs, they don't really even have a hip, but they do have some remnant hip bones, and that is thought to be from an ancestor in the past, common ancestor, that did have 4 legs. Eye remnants buried under scales in blind species of fish, and so obviously they have no use for eyes, but probably an ancestral species did have eyes, and so now they have some kind of remnant of it. This is what we call homologous structure. That means that they have different functions, but they have the same structure. Okay, so remember, home means same, so same structure, not necessarily function. So, for example, the human arm is for balance and walking and grabbing and reaching, and the cat walks on their forelimbs, and the whale swims, and the bat flies. So all different functions. But if you look at the physical structure, you can see that this bone is homologous here, and then you have these two, and then right there you can see them. And then you have the what we call the carpals here, right there, right there, right there. They might be even a little bit different shape, but they're still in that basic same order. Then we have the metacarpals, And then we have the phalanges. And again, our phalanges are kind of medium-sized, really long, and also pretty long here. So they are different, but at the same time, they have that same basic structure. Different function, same structure. An embryo shift to a human, very, very similar. These are some vestigial structures that actually don't develop in the human embryo, like the tail here. This actually, apoptosis reduces that. But that's where you get your tailbone, your remnant, which is also a vestigial structure. And then pharyngeal pouches are here in the chicken in the human embryo, but we don't have those as adults or when we were born. Other evidence, molecular similarities. So all forms of life have the same genetic language or code, either DNA and RNA. Every language is made up of these four nitrogen bases. It is likely that all species descended from a common ancestor. Even bacteria and humans share some genes in common, which is hard for people to believe, but we share genes in common with yeast and bacteria and everything else. Think about it like this. All living things do cellular respiration, right? Well, cellular respiration, there are genes that encode the ability to be able to do that, to create the parts of the cellular respiration cycle, to create that process and how it happens. And if we all do it, then we all have to have something there that's in common. Remember, your genes make up everything about you. And even though we are very different than, say, a pig, there are a lot of things that are very similar. We are very different from a bacteria, but there are still some things that are very similar. An evolutionary tree is a diagram that reflects evolutionary relationships among groups of organisms. Typically, we use a lot of DNA evidence now because we have the ability to do that. And you're going to do a lab on Wednesday and Thursday this week that uses that same BLAST system. If you remember when we did bioinformatics, we're going to use BLAST to compare DNA sequences from different species and kind of try to create an evolutionary tree based on that. So here is an example of an evolutionary tree. There are lots of different types. This is just one. These branching points, these are going to represent a common ancestor. Okay? So the more recent, so in time, when you look at this kind of the scale of time, this is going to be more recent and older, like more distant from the past. I guess this is probably the word I should have used. So ostriches and hawks and other birds right here, these guys share a common ancestor more recently than, say, lungfishes and hawks. Their common ancestor doesn't branch back until back here, which is a much, much further back in time. So you will find that the closer, more recent in time that they have a common ancestor or a branch point, the more closely they are related. These markers indicate characteristics that exist in anything on this side, on the right side of that marker. So here, the only thing in this cladogram, or this evolutionary tree, whatever you want to call it, the only thing that has feathers are going to be the ostriches and the hawks. These guys do not. If you go over here, amnion, this is going to be part of their early development, and anything on this side of is going to have an amnion as they develop. Digit-bearing limbs, so multiple fingers or limbs, digit-bearing, that's how you can say your fingers. Anything on this side is going to have that. So like the lungfishes don't have the digit-bearing because they're not on this side of that marker. And that's how those work. So we're going to have a chance to create a cladogram, and you'll have to analyze some cladograms. Cladograms will be on the AP exam. I can guarantee it somehow. You may have to create one. You may have to analyze one. So we're going to do some practice of those. I wish we had some more time to do that. Convergent evolution, independent evolution of similar features in different lineages. Some things evolve because of similar environments rather than a common ancestor. We have to make sure that we are not making inferences that are not necessarily true. This is where DNA comes in. So even though they may have similar features, if DNA genetically is not very similar, then they may not actually have a shared common ancestor, or at least not a recently shared common ancestor. So we have to look at more than just do they have legs. Okay, we have to think about what the structure of that leg is or what its environment was, because obviously different populations are going to develop according to their environment. We learned that. That's natural selection. Analogous structures, remember that word, are going to be structures that are because of convergent evolution, not indicative of a common ancestor. Homologous structures indicate a common ancestor and is responsible for the same physical structure, though function may be different. Analogous indicates the environment was the same, which gave way to similar functions, but not structure, and is not indicative of common ancestry. So if you remember anything here, homologous is indicative of common ancestry. Analogous is not. The fossil record documents the patterns of evolution, showing that past organisms differ from present-day organisms and that many species have become extinct. Fossil records also show changes in groups of organisms that can shed light on the origins of new groups of organisms. So this is why we know about past, because we weren't there. And so fossils are a way to document and to record the past. Think about the sticklebacks. The only way to know what the population was doing in the past was to look for the fossils of it. Okay, here is another phylogenetic tree, cladogram, whatever you want to call it. You've got your species over here, and this is showing the key for the different parts. Notice that living cetaceans, I'm not sure I'm pronouncing it right, only have this pelvis. But obviously they don't have any legs, right? There's no legs on a whale. But they still have a remnant of a pelvis. But remember, that is a vestigial structure. And then here's the common ancestor. Remember, these are the branch points. So if you look at these are more commonly, more recently related. And then more distantly related would be things like these two on the outside. Biogeography is the distribution of species geographically. Continental drift, hopefully you've heard of that somewhere in the past, can explain how some species resemble individuals in other areas, as well as fossils found in different areas. Pangea was when all of Earth's land mass is united in one large continent. I'll show you some pictures in a minute. Islands generally have many species of plants and animals that are endemic or found only in those areas, because islands are more recently created. So here is the idea of Pangea, and so they think that what happened was this was one large landmass, and then it spread away, and they know that because they really can almost puzzle piece these together, especially with, if you look at the plate tectonics here in a second. The fossil record is based primarily on the sequence in which fossils have accumulated in sedimentary rock layers called the strata. Some useful evidence comes from insects preserved in amber, or fossilized tree sap, and mammals that have been frozen in ice. The fossil record is an incomplete record. Okay, that's what's important here, is it's incomplete. And so that makes the past a little bit of a difficult reconstruction. It's kind of like putting together a puzzle and missing a bunch of pieces, but still trying to figure out what the picture looks like. But they are a chronicle or a documentation of evolutionary change. The fossil record is biased in favor of species that existed for a long time. So obviously, if the fossil, or if the species never created a fossil, then it's going to be really, really hard to know anything about that, because we weren't there and we don't have that information. So that's where the incomplete, and then it's biased because we do have the abundant species, because usually they created the fossils. And it's also biased for certain kinds of animals, things that had hard shells, skeletons, or other parts that actually help with the fossilization. So there's a lot of things we don't know because they didn't create fossils. Gaps in the fossil record continue to be filled by new discoveries. This is not a old science that doesn't happen anymore. We still have paleontology. The order of the fossils in the rock layer only tells us relative ages. One is older than the other, but it does not tell you absolute ages. Absolute dating is through radiometric dating, but that does not mean that it's errorless. Okay, that means that there is still some error. It means that if given in years rather than before after. So you can say this fossil is X number of years old. That doesn't mean that's errorless. Okay, you could be off by several thousand years. Radioactive dating is based on the decay of radioactive isotopes. And so that's how we can kind of put these guys in order and actually give them a time frame. A little bit about radioactive dating, but I don't think you're going to be asked anything about this. Basically, the scientists have figured out when you look at one isotope and they figure out of a daughter isotope what the half-life is, so how long it takes for that to basically decay. For example, carbon-14 has a half-life of 5,730 years, and uranium has a half-life of 4.5 billion years. Each radioactive isotope has characteristic half-life, which is not affected by temperature, pressure, or other environmental factors, which is why it's used for this purpose. Fossils contain isotopes of elements that accumulated in the organism when they were alive. After the organism dies, the radioactive isotope starts to decay, and then we can measure how much decay so that we can determine the fossil's age. The method works best for fossils up to about 75,000 years old. Older fossils contain too little C4 to detect with current techniques, so they have to use different isotopes. Sometimes rock layers are not clear, and scientists must infer age based on isotopes of the surrounding environment. And this is just showing a diagram of the half-life of that. Life history. Fossils have helped scientists create a geological record of life history, or Earth's history, sorry. Scientists have divided time into eras, and eras are usually divided by major extinction events seen in a fossil record, when many forms of life disappeared and were replaced by forms of life that evolved from the survivors. So that's how they define an extinction. And this just shows kind of what they've looked at as far as time periods and how long ago they were. Rise and fall of major groups of organisms has helped shape Earth's history. Plate tectonics. The continents are a part of a greater plate on Earth's crust that essentially float on the mantle. Movement of the mantle causes these plates to move over time. That's continental drift. I showed you that earlier. It's also what causes earthquakes when they move. Continental drift alters the habitats in which organisms live. Climate changes result when a continent shifts its location. Continental drift also promotes allopatric speciation, remember where they're geographically isolated. And it solves the puzzle about geographic distribution of extinct organisms. For example, you might find species that are right here, fossils right here being the exact same as fossils right here. Well, that makes sense because actually so many billion years ago, these two were the same piece of land, if that makes sense. Mass extinctions, they have significant and long-term effects. The fossil record indicates it takes about 5 to 10 million years for the diversity to recover from a previous mass extinction. It can alter the communities by changing the types of organisms in them. Adaptive radiations, periods of evolutionary change in which groups of organisms form many new species, whose adaptations allow them to fill different ecological roles or niches in their communities. Large-scale adaptive radiations occurred after the 5 big mass extinctions. Fossil evidence indicates that mammals underwent a dramatic adaptive radiation after the extinction of the dinosaurs. With the disappearance of the dinosaurs, mammals expanded greatly in diversity and size because they don't have those big, large predators anymore, and they filled the niches the dinosaurs once occupied. And this just shows you a graph of some of the mass extinctions. The extinction rate, you can see it, huge extinction rate right there. Where is that? Scientists also want to understand the biological mechanisms that underlie the changes in the fossil record. We look at the genetic changes, specifically genes that influence development. Heterochrony is the evolutionary change in the rate or timing of developmental events. And homeotic genes, we introduced this some time ago, genes that control the placement and spatial organization of body parts. Hox genes provide positional information in an animal embryo. So where's the head going to go, where's the tail going to go? In snakes, where changes in 2 Hox genes were expressed, that suppressed the actual limb formation. Think about some of those videos we watched, and they talked about being able to actually find the gene in the stickleback fish that suppressed their spines. It's the same idea. Oops. And then you will see that we can also compare embryo information. So, for example, a human fetus, human adult size head, very different from the chimp, but if you look at the fetus, they're actually fairly similar. Novel and complex biological structures can evolve through a series of incremental modifications. Remember that is key here, incremental modifications, things that you wouldn't even notice in your lifetime. but over a million years, they accumulate, and that means a big difference, each of which benefits the organism that possesses it. Evolutionary trends result from the interactions between organisms and their current environment. Natural selection. The origin of life. Experiments have shown that it is possible, this is a big word here, possible to produce organic molecules, so things like carbohydrates, proteins, nucleic acids, needed for life from inorganic things, because we think that Earth's atmosphere contains like nitrogen and hydrogen and things like that. Those are inorganic, and we were able to, as experiments have showed, that it is possible to produce organic things, things that are needed for life, from those inorganic molecules. Organic molecules can spontaneously assemble into protocells, or membrane-bound droplets that have some properties of cells, not fully cells. The endosymbiont theory, we learned that early in the year. It talks about how mitochondria and chloroplasts are really like, they're almost like little functional cells inside the cell. And so they think that these were engulfed by cells, and that was a positive relationship, so it continued. The first genetic material may have been RNA. So this is the idea that endosymbiont theory, that you just have this plasma membrane protocell type thing, ancestral prokaryotes, what they call it. And then the photosynthetic prokaryote, and then the mitochondrion were separate, and then they somehow made their way into cells, and that became a really good relationship, and so it progressed because they survived really well with those extra parts. Phylogeny, this is a huge focus that will be on the AP exam. That's the evolutionary history of a species or a group of species. Systematics is a discipline focused on classifying organisms and determining their evolutionary relationship. We use data from ranging from fossils to genes to infer relationships. And this is a changing field of science. It's not always the same. Taxonomy is kind of a way of classifying things. Not all scientists agree on how things are classified. Binomial is a two-part naming system, genus and species. For example, Homo sapiens. First letter is capitalized, and the second one is lowercase, all in italics or in quotes if you're writing it. Classifying is our way to structure our human view of the world. Organisms are classified into categories. This is the most inclusive domain, so we're in the same domain as every other animal on the planet, and actually any other planet, because the domain is either bacteria, archaeobacteria, or eukaryotes. And then they get classified, and then this is the least inclusive. So the only group in the human category of species is Homo sapiens. But if we go up in order, we can be all the way up to domain. We are classified along with every other eukaryotic organism. And this just shows you another graphic of that. A phylogenetic tree is an evolutionary history of a group of organisms that can be represented in a branching diagram. The branching patterns often match the classification system. It's representative of a hypothesis about the evolutionary relationship. Each branch point represents the divergence of two evolutionary lineages from a common ancestor. So here you go. And I talked about the common ancestor, common ancestor, common ancestor. This is the oldest common ancestor of all of these in this particular diagram. Here's another way you might see it. Again, this is the common ancestor of all of these. This is the common ancestor of just these two. What we can learn, it's intended to show patterns of descent, not similarities in their phenotype or their physical looks. Sequence of branching in a tree does not necessarily indicate the actual ages of the particular species. Unless given information about the branch length, they are not proportional to time. Don't assume that a taxon in a group and a tree evolved from the taxon next to it, only that the lineages leading the two taxons evolved from the same ancestor that is now extinct. So if we go back and look at this, this does not mean A came from B or B came from C. This means that B and C share a common ancestor here that is probably not currently living, and that A and C share a common ancestor back here that is, again, probably currently not living. Homologies are similarities due to shared ancestry. The number and arrangements of the bones or forelimbs of the mammal, we talked about that. genes in two organisms share many portions of a nucleotide sequence, it is likely that their genes are homologous. We're going to look at this in our blast lab. Analogy are similarities due to similar environments, not indicative of common ancestry. North American and Australian mole actually are not very closely related, but they have a lot of similar characteristics because of their environment. DNA and RNA sequences in distantly related species usually have different bases at many sites and have different lengths because insertions and deletions have accumulated over time. Researchers have developed computer programs that estimate the best way to align comparable DNA sequences segments differing lengths. So we use NCBI and we use their BLAST comparison. Molecular systematics is a discipline that uses data from DNA and other molecules to determine evolutionary relationships. This is how most evolutionary relationships are determined because we have the ability to do DNA evidence. Now that may not be necessarily possible with us. And this is just showing you how DNA can change. Cladistics is a common is where common ancestry is a primary criterion used to classify organisms. Species are placed into groups called clades, which include an ancestral species and all of the descendants. The clades are nested into larger clades like taxons. The shared ancestral character, or the character that originated in the ancestor of the taxon, is noted like a backbone. I'll show you that in a minute. Shared derived characters are an evolutionary novelty unique to the clade. That's what we call a shared derived character, and I'll show you in a minute. Found in mammals, but not in their ancestors. Hair, for example, will be a shared derived character. So we would put like maybe a shared derived character here or here, which means that anything over here would have that character, but not this organism. Shared derived characters are unique to particular clades. An outgroup is a species or group of species from the evolutionary lineage that is not known to have diverged before the lineage that includes the species being studied. The in-group is a species that you're actually studying. By comparing members of the in-group with each other and with the out-group, we can determine which characters were derived from the various branch points of vertebrate evolution. There we go. So, hair, something only this one has. Amnion, something that both of these have. Four walking legs, something that all of these have, but none of these have. And you can see that. This is a table. You're going to fill something out similar to this in your lab, except you're using genetic information. Here they're talking about a 1 indicates that information is, they actually share that information. A 0 means that they don't. And so you just kind of use this, and then you can say, okay, well, most of them have this. And so we're going to put this the furthest in time, or more distantly in the past. And then most of them share this. This is the next thing most of them share in common. So we're going to put that here. And then this is the next one. And you just keep working your way down until you get down to, oh, this organism is the only one with this particular derived character. In some diagrams, branch lengths are proportional to the amount of evolutionary change or to the times in which particular events occurred. Don't assume that unless something indicates that. For example, in this one, if it indicates this, indicates time, and then this is more recent in time, so don't assume branch length means anything unless it tells you it does. Here's an example where it actually tells you the information about time here. Systematics can never be sure of finding the most accurate tree, but they can narrow the possibilities by applying the principles of maximum parsimony and maximum likelihood. Maximum parsimony means they investigate the simplest explanation, and maximum likelihood given certain probability rules about how DNA sequences change over time. It's kind of like the most likely sequence of evolutionary events. Scientists have developed large computer systems that search for trees based on these two ideas. That's something our brains are not accustomed to doing. Okay, so this example is showing that these two diagrams are both possible, but more probable would be this one here. And so there's whole computer programs designed to identify trees and say, okay, which one's the most probable. Any phylogenetic tree represents a hypothesis about how various organisms in the tree are related to one another. Phylogenetic trees can be modified when new evidence is presented. Scientists make and test predictions based on the assumption that the tree is created is correct, and then they might find evidence that says it's not, and then they just modify it. So phylogenetic trees are not set in stone. Okay, they are constantly being modified. Molecular biology has helped systematics. Different genes evolve at different rates. Orthologist genes are found in different species, and their divergence traces back to the speciation events that produce the genes. Paralogous genes are homology results from gene duplication, since multiple copies of these genes have diverged from one another within a species. And this just shows that. I'm not really sure you need to know much about those phenotypes of genes, so I'm going to skip over this for some time. Molecular clock is like a yardstick for measuring the absolute time of evolutionary change based on the observations that some genes and other regions of genomes appear to evolve at constant rates. The assumption is that the number of nucleotide substitutions in orthologous genes is proportional to the time that is elapsed since the species branched from a common ancestor. In the case of paralogous genes, the number of substitutions is proportional to the time since the ancestral gene was duplicated. And here's what we get. Number of mutations, divergence over time, and we kind of put a nice little line of best fit in there. We've done that to actually adjust and account for uncertain information about HIV and when it actually probably first appeared. We use all this information, we do a line of best fit, and it actually gets us back to probably mid-30s. Scientists graph the number of genetic differences against dates of evolutionary branch points that are known from the fossil record. No gene marks time with complete precision. Some parts of the genome appear to have evolved in irregular bursts that are not clockwork. So one of the big downfalls of something like this is we assume a constant rate, which is not necessarily the case. New information. Phylogeny based on genetic data give rise to a change in the way we classify. We're now doing a lot based on DNA. Domain was added because some bacteria were not like others so much that they were given a whole new classification. So we separate archaea and eubacteria for that reason. Archaea is a diverse group of prokaryotic organisms that inhabit a wide variety of environments, like really salty, really hot environments, anything that's just abnormal. Bacteria differ from archaea in many structural, biochemical, and physiological characteristics. Eukarya is anything that contains a true nucleus. And these are the domains here. These are really kind of a cool setup. They live in really bizarre environments that we thought nothing could live in before. Comparisons of genomes from the three domains show that there have been substantial movements of genes between organisms in the different domains. Horizontal gene transfer is a process in which genes are transferred from one genome to another through mechanisms such as exchange of transposable elements and plasmids, viral infections, and perhaps the fusion of organisms. Yeah, so eukarya is here, archaea here, but they do share some things in common, which suggests some horizontal gene transfer. History of life. Scientists continue to debate whether early steps in the history of life are best represented as a tree, a ring, or a tangle's web. Ring is one that I just thought was really interesting, but it is a constantly evolving science. And we will talk more about the biggest part of this, which is cladograms. Okay.